Welcome. Hi, on this really beautiful spring day, sunny outside, systems biology inside. And let's all start by invite you to take a nice deep sigh of relief, as we always do. So this is the third lecture in the course. And so far we looked at transcriptional networks. And we noticed that they're made of perhaps it's a good time to clean out the board. Take a nice deep sigh of relief in your honor as you find a seat. Is made of interacting proteins and genes, and we saw that we write like this: when transcription factor X binds the DNA and changes the production rate of Y, it can increase the production rate of Y, or if it decreases the production rate of Y, we write it like this. And X, by the way, represents a kind of molecule. There could be 100 copies of this transcription factor, or 1,000 copies of this transcription factor. And Y represents a different kind of protein. We analyze what happens when X affects its own production. In fact, what we did was we took a look at the network and we noticed that there's recurring patterns. And we call them network motifs. Those recurring patterns occur with many, the same interaction pattern occurs with many different proteins. And this negative autoregulation is a network motif. Occurs again and again, again and again and again and again, more often than we might expect by chance. And we saw that it has specific functions that it can do. These functions are built into the wiring. They don't depend so much on the parameters, the rates, etc., the numbers on the arrows. For example, we saw that negative autoregulation can speed the response time and make the X concentration robust with respect to fluctuations in production removal rates. And today we're going to look at another larger font. Yeah, thank you. Today we'll look at a more complicated circuit that's a network motif made of 3 genes, you can see. And we'll analyze its function. So conceptually, it will be a very similar design to the last lecture. But you will learn about a new circuit. And we're going to do the same thing next lecture too, and then move on to a different topic. Okay? Ready? Yes? Question?
We talked over there to talk about positive autoregulation.
Ah, positive autoregulation. So this is also, it's also a network motif. I think it was in the exercises. Has anybody here looked at
It wasn't the exercises.
it was a bit difficult in the exercises. Yeah. So yeah, we can talk about positive autoregulation. Ah, welcome. Let's take a nice deep sigh of relief in your honor. Why don't you find a place? There's places here. Yeah, and positive autoregulation. So, you know, if negative autoregulation speeds response times and makes X steady state robust with respect to fluctuations, positive autoregulation does the opposite. slows response times and makes X steady state more variable between cells. Yeah. In certain extreme cases, it can even make the population be all or none, either making a lot of X or none of X. If you want to find a place to sit, I warmly recommend. Then there's two places here. There's a couple of places there. Because in a collective situation, if one person is uncomfortable, everybody feels it somehow. Biological examples of negative autoregulation, for example. In bacteria, there's a transcription factor, a repressor, that represses its own production and also represses DNA repair proteins. So normally, it just shuts off DNA repair proteins, and there's no need to repair the DNA. But if there is DNA damage, the cell has ways to find out about that. A particular protein called REC-A binds the DNA and it physically cuts this repressor. So this is the repressor level, this is time. If there's DNA damage, within seconds, protein is cut. And what happens to the DNA repair genes that it represses? It's no longer present. So its repressive action is no longer present. And therefore, they get expressed. And DNA is repaired. Right? That's the way it works. So the DNA repair proteins, function of time, start being made. And they repair the DNA. And when they repair the DNA, there's no longer damage, so this repressor is made again. And when it's made again, so it has some threshold, right, for repressing those proteins. When it reaches high above its repression threshold, then these are not made anymore, and they get diluted away. But the function of this negative autoregulation, as we said before, is to speed things up. So this negative autoregulation does this. So that in fact, negative autoregulation helps E. coli recover faster from DNA damage. In particular, one of these proteins tells the cell to stop dividing, because E. coli doesn't, and our cells too, when you get DNA damage, it doesn't keep dividing. That's one way to, in our body, that protects us from cancer. Also, E. coli stops dividing. It becomes filamentous cells. So if you want to recover quickly from DNA damage and keep dividing, you'd like this repressor to come up quickly, so it'll shut down those proteins for DNA repair and for recovery. So that's one example where experimentally was shown that if you mutate away the repressive binding site of LEX-A and its own promoter, you get a slower recovery. Did I explain myself? This is a biological example. I know that people have been asking for a few more examples. And positive autoregulation happens in situations where you need. Well, actually, we'll see one today, so we'll talk about it. Okay, so that's our review. And now let's look at our complicated graph that describes the E. coli transcription network. And we'll search for patterns. If we search for special patterns that have 2 genes, we don't find anything special. Find V's, one transcription factor that regulates 2 genes, or 2 transcription factors that regulate 1 gene, things like that. If you look for 3 node patterns, we do find something interesting. So before we consider, let's write down all the possible ways to connect 3 genes. Right? So there's 13 ways to connect 3 genes. 13 directed, connected subgraphs of size 3. I'll just try to draw. So these are the 3 genes. So this is one possibility. Right? This transcription factor regulates these 2 genes. And here's another possibility. I'm not even going to draw the points anymore. You understand the arrows, right? Second possibility. OK. And of course, we can have a cascade. Well, let's continue. You can have a mutual situation. So you can have something like this. They regulate each other and the 3rd gene. And we can have something like this. You see, they're different because here, this guy who participates in the mutual regulation regulates, and here they get regulated. Topologically different. And we can have, of course, this situation. So we have 1, 2, 3, 4, 5, 6. All right. And what about this? And this? This is a cycle. This is not a cycle. And this. 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 These are the 13 ways. Maybe is something not clear here the way I drew it? Clear? So this is everyone regulates everyone else. This happens actually in stem cells with 3 transcription factors, control pluripotency, but not in E. coli. Yeah?
Why don't you include autoregulation?
Oh, great question. Yeah, thank you. So on top of this, I can include autoregulation or not autoregulation. When you do that, it grows to 30. There's more possibilities. But let's not include autoregulation for now. We'll add it later. That's a lot of combinations, right? So that's why complex networks are so complex. You have, when you try to think about 4 genes, there's 199 possibilities. 5 genes is 5,000 and something possibilities. It grows exponentially. And in principle, all of them can occur. So we could be faced with a situation where this network contains many kinds of small circuits. But when we compare this network to randomized networks in different ways, it turns out that only one of these occurs more often than you expected by chance. And in fact, very few of them occur at all in E. coli. And only one of them occurs it's significantly large numbers. In E. coli, you get this one, and this one, and this one, and this one. Feed-forward loop. This one, the cycle, this feedback loop, is an anti-motif. Occurs less than expected. Actually, it occurs zero times. And this one occurs about 100 times, each time with different x, y, and z. So it's 7 standard deviations more than you expect by chance with very stringent random networks that can keep the same number of nodes and arrows, and even distribution of outgoing arrows and incoming arrows. Yeah.
By the arrows here, you mean activators and repressors?
Yeah. So these arrows, right now I drew, this could mean activator and repressor. Soon we'll analyze. Thanks. This could be either sign right now, the way I'm drawing. So if we add activators and repressors, we have many, many more possibilities, of course. In this lecture, we're going to study this feed-forward loop, right? So it is more than seven standard deviations away from randomized networks. And it is a network motif. Okay, so we're going to try to understand this circuit. And maybe you think, what's wrong with the feedback loop? Why isn't it? Why doesn't it appear? So that's our program, 3-node patterns. And the feed-forward loop is a network motif. One moment.
All the rest will show as we expect from a randomized network.
Can you repeat the question?
All the rest 11 cases.
What about the other 11 cases? So they don't appear at all. And these appear quite often, about as much as you would expect from a network of this size. And actually, we'll understand them. As you notice, all three of these are part of this. Okay, there's a cascade, so we can understand them. You'll understand by the end of the class. You'll understand them. I hope they're dynamic.
You call all the zeros anti-motif?
Well, this is very rare in the networks of this size, for example. So I don't know, but this you should expect a few of them, but you don't see them. But from my point of view, like I said last time, another opportunity for a nice deep sigh of relief. Welcome. The more you do, the better. These sighs of relief. So everything that appears, even one time, is interesting, because it has to be selected against evolutionary forces, and it's worth studying. But in the case of the E. coli network, you see how simple it is. There's only these subgraphs at all. So it's actually kind of directed acyclic graph in computer science with very few kinds of subgraphs. All right, so now let's get into Eitan's question about the signs on the arrows, plus, minus. Because there's another, so these, if we look at this pattern, X regulates the production rate of Y, and Y regulates the production rate of Z, and X also directly regulates the production rate of Z. So if you look at this pattern, what you see is that X, this transcription factor, regulates Z in two ways, directly and through the friend Y. So a direct path and a delayed path. Okay? That's the feed-forward loop. There's a direct path, and there is a delayed path that converge on the same target. Now, each arrow can be positive or negative, even though here I drew the arrows generally, like it could be either positive or negative, right? So, and there, as I told you, there's about 50-50 plus and minus in E. coli. So now we can ask how many types of feed-forward loops there are. There are 8 possible types of feed-forward. From now on, I'll use FFL to say feed-forward loop. 8 possible types, because each arrow can be plus or minus. 2 times 2 times 2, 3 arrows is 8. So let's write them down. So it could be that X activates Y, that is, increases the production rate of Y, Y activates Z, and X activates Z. Is this big enough for the camera? Almost. Yeah. And this we call coherent type 1, or C1 feed-forward loop. And why do we call it coherent? Because X activates, and activates also by activating an activator. Okay. On the other hand, we can have a situation where X activates Z and activates a repressor of Z. Right? This is a repressor. So here it's strange. It's activating with one hand and activating a repressor, which is actually inhibiting with the other hand. So this is why we call this an incoherent type 1 feed-forward loop, I1 FFL. Okay? These are 2 possibilities, and there are 8 possibilities in total. Right? So I'll try to draw the coherent ones. So X can repress Z, and at the same time repress an activator of Z, or X can repress Z, and at the same time activate a repressor of Z, or X can activate Z, and at the same time repress a repressor of Z. This is also activating. These are the 4 coherent types. And I don't remember by heart how we named them. This is type 2. This is type 3. This is type 4. Okay. And then there are 4 incoherent types. So we can try to draw them. There's X can repress Z, but repress a repressor of Z, right? So it's repressing here and activating here. Minus minus is plus. And X can, this is fun, repress Z and activate an activator of Z. Or X can activate Z and activate, or and repress an activator of Z. So here it's doing something bad to something good. It's like so we call this this one type 2, I'm sure, and type 3, type 4. Okay, so these are the 8 possible types, right? And we might expect that they occur equally, more or less, because there is equal number of plus and minus signs. But in fact, they don't occur equally. When you do the counting, you see that only 2 of them occur. Let's say 90% of the feed-forward loops in E. coli are of 2 of these 8 types. 90% of E. coli FFLs are coherent type 1 and incoherent type 1. This one and this one. And the other ones occur, but much more rarely. And when other labs, our lab and other labs, looked at other organisms like yeast, again, out of the 13 possible 3-node patterns, the feed-forward loop is the only one that's significant and occurs massively hundreds of times. And of the 8 kinds of feed-forward loops, only the coherent type 1 and the incoherent type 1, where they stand out an order of magnitude more than the others. And when you look at human cells, mouse cells, you begin to see more of these other types. And sometimes these repressors are microRNAs, for example. But these types are the dominant types. It's dominant. Why I mean dominant? This is interesting use of language. What I actually mean is abundant. Right? Not always the abundant is the dominant. All right, so we have the 8 types and this idea of coherent and incoherent circuits that can do the same thing or opposite things. So what else do we need to know about the feed-forward loop in order to try to understand it? Just to state what I said again, there's a great simplicity from this point of view in the E. coli transcription network. Instead of having to be worried about 13 possible ways to connect genes, we only have to basically be worried about one kind. And even that one kind, instead of being concerned with 8 types, we need only to think about 2 to a first approximation. And this kind of simplicity is a great thing if you are hopeful about the potential of human beings to understand biological networks, because it didn't have to be this way in principle. And we'll try to see if we can understand the dynamics of these circuits. We're actually going to focus on this one this time. And we're also going to understand this one. Now, we need to understand one more thing before we continue. So, I'm going to erase. You notice that in this circuit, there's two arrows that enter one gene. And that's interesting. That means two transcription factors need to regulate the production rate of Z. And this raises the question, how do they work together? So X and Y regulate Z production rate together so that its production rate is a function of the active concentration of X and Y. Remember that X and Y are proteins, could be in active or inactive conformations, and the external signals make them active and inactive. So we can ask, what are the possible ways in biology that two transcription factors work together? This, we call this object the input function of Z. This two-dimensional function, this function of two inputs. I just want to now just kind of zoom into the molecular details just for a second to explain in another way what we mean by these arrows, even though by now I'm sure most of you can do this by heart. So X is a transcription factor that transits in microseconds between active and inactive conformations, and a biological signal pushes it to be active. X. Now X binds this piece of DNA we call the promoter of gene Y at a specific site to change the production rate of Y. X also binds our output gene in this site. It has some site here that's very similar to this site. The site is, of course, a sequence of letters that X star recognizes. And Y binds its own site with polka dots, let's say.
But it also shifts from
Oh,
active
thank you. See. And it has a signal SY that pushes it. Exactly, it has its own signal. Does it have to have its own signal? In E. coli, as far as we know, all the transcription factors have a signal. Sometimes we don't know what it is. But in our cells, it's possible to have transcription factors that are computational nodes. They're made and they don't have a signal. They act as elements in a computation. But generally, I would say yes. This signal could be a sugar from the outside. It could be another protein binding it. Only when that protein gets phosphorylated, that protein comes off and the transcription factor is active. Something that keeps the transcription factor out of the nucleus. It could be many things. But the point is that it acts quickly. And these time scales are the slow time scales on which we're working on. So all these reactions are fast. These are the slow time scales. Some other molecular details that are important for us is that this transcription factor Y has a certain threshold for binding to the site. So this is the concentration of Y star needed to actually activate or repress that gene. Ky is the concentration of Y star needed to activate or repress whatever Z promoter. And of course, there's a Kx Y here. This is, I'm going to call it Kyz. Actually, to be precise, it's the concentration of Y needed to repress the Z promoter. And this is Kxy, the concentration of X needed to repress or activate Z. So you see, we need some numbers. You can measure those numbers. And evolution can change those numbers by changing these letters. So that if the binding site isn't the perfect set of letters, but different by 1, you'll have weaker binding, which means higher K. You need more of the protein in order to affect this. And then there's the question I asked, how do X and Y, you see they have 2 sites or sometimes many sites together on this promoter. How do they work together? And you can measure this, of course, experimentally by making the cell produce a given amount of X and a given amount of Y and measuring the output Z, let's say by using green fluorescent protein like I mentioned last time. And in general, these functions are often the input functions resemble an AND gate. Both X star and Y star are needed. And OR gates. Either one of them is enough. And in fact, they're continuous functions. If I plot them as a function of X star and Y star, they look like an AND gate has a mountain here when X star and Y star are high. An OR gate has this ridge when either one of them is high. And you can see kind of variations on this theme. It's very rare to get other things like people here who know about Boolean logic, like exclusive OR or things like that, exist, but it's rare. Where exclusive OR means that X can activate, Y can activate, but when they're both, they don't activate. Something like that. That can happen, but it's rare. So these class of monotonic input functions. So not only do we have, in principle, 8 types of feed-forward loops. Each one can be either an AND gate or an OR gate. So we can have X, Y. This is the way we write it. This is with an AND gate. And this is the coherent type 1 feed-forward loop with an OR gate, you see? And these symbols are come, of course, from Boolean logic or electronic engineering. So each one of the types can be at least with these either AND or OR gate, and we'll see that plays a role too. Great. Good. That's again. And we're actually ready to understand the dynamics of the feed-forward loop right now. We're going to do that right now. Just to restate the premise or the main idea in the course is that when we find the biological structure, we dare to ask why. Why is it there? Our answer is based on the only theory we have in biology, which is evolution. It's there because it confers a benefit to the fitness of the organism. In case of network motifs, we think a computation that this circuit can do a computation that simpler circuits cannot do, which is important. It helps the bacterium or the organism make more babies, survive better, and therefore it's worthwhile keeping this circuit against the mutational forces that would tend to quickly destroy structures. In general, mutations destroy things. It's easier to destroy than to build, as you know. So when we see a structure, we think about why. And it's unusual to ask why in biology, molecular biology, a lot of times. But we dare to here, and answer with kind of mathematical language. And in the end, we end up with design principles, which are concepts that unite different systems. Okay, so we're going to ask why there's a coherent type 1 feed-forward loop. And we're going to even use an AND gate now, just for this demonstration. And we'll try to show that it acts as a filter that responds only to persistent signals and shuts off rapidly. So we're going to do the following. So the circuit we're dealing with here is the coherent type 1 feed-forward loop with AND logic, because we need to start somewhere, right? And we notice that there's a signal, SX, this signal that activates X. I forgot to write Y here. And there's a signal, SY, that activates Y. And because it's a coherent type 1 feed-forward loop, all of the X and Y are both activators. They increase the production rate of Z. And we're going to analyze the situation when we assume, one, that SY is present. So we have the signal. Whatever Y, protein Y is made, it quickly becomes active. And we're going to also do, we're going to look at sudden changes in the input signal, Sx. We're going to look at on steps in which Sx as a function of time goes up, and off steps where it goes down. Again, just to make the analysis simple so that you can understand, you can explain it very clearly. Of course, you can analyze this for any time-varying signal with the equations that you already know how to write, production and degradation equations. But this is simple enough to solve graphically. I'm going to draw 3 graphs here. This will be the input signal. This will be the concentration of Y. And this will be the output concentration. So here's our on step. We're going to assume that if, for instance, Sx is a sugar, the bacteria swims into an area that has a lot of sugar. And of course, X becomes active, and X star rises very quickly on the timescales we're concerned with. So we have suddenly this is activated. Now what happens to Y? Before we had the sugar, or the signal, we couldn't make Y. So it was 0. But now that we have this signal, X becomes activated, quickly goes to the promoter, Y starts to be transcribed, translated. Y rises. Y rises according to the dynamics that we know, this exponential dynamics, to approach its steady state. What about Z? Z was 0. Now at time t equals 0, X is active. Does Z start being produced? I hear the answer no. Why not? Because it's an AND gate. You also need Y. So it's not produced. When does it start being produced? The answer here that I heard is when Y star exceeds the activation threshold. K, Y, Z. And at this time here, Z starts being made. Therefore, what we see here is a delay. So there is a delay after on steps, and just very intuitively, we have an AND gate between an immediate and a delayed response. This AND gate waits for both of them, therefore there is a delay. The timescale of this delay, by the way, in order to find this delay, we notice that Y we have our regular equation that you're familiar with. And we want to ask, where does this equal the threshold? That's this delay time, the time when it equals the threshold. And when you try to solve this thing, and if you take the logarithm on both sides, you find a solution that this delay is equal to 1 over alpha y log 1 over 1 minus k yz over y steady state. So you can, the point here I want to make is this delay can be tuned by evolution by playing with any of the parameters of protein y, by playing with its threshold kyz, by playing with its steady state level, or with its the removal rate alpha y. You can tune this delay. You can plot this function. It's an interesting function. In real systems, it's usually something on the order of 1/10 of a generation. Or in E. coli, let's say 10 minutes, 20 minutes delay. This is the order of magnitude. You can make it an hour by playing with the parameters. For example, if you can analyze this anyway. If we increase the threshold, you should get the delay is longer. If we decrease the threshold, the delay is shorter, etc.
Generation time for equal is 20 minutes.
So the generation time for equal is 20 minutes. That's like the world record, but usually in the lab it's 45 minutes, 30 minutes, 60 minutes. Now let's what happens with an off step? Yeah.
About the end thing, okay. You don't really need a direct space in order to get the delay time.
The thing is, you don't need an AND gate in order to get the delay time. A cascade would do it.
Yeah.
So let's look at the off step. So we need both of them. So let's see the off step. So please see if you can follow. Off step is this situation. Y star, which was high. Now, when there's no X star, Y starts being just removed, right? It just goes down. And what happens to Z?
Shuts down immediately.
Okay, somebody here said shuts down immediately. Why is that? Because of the AND gate, it's only enough that one of the two inputs goes away, because you need both. So there's no delay here. When X star goes away, because X star transits back to X, this takes milliseconds, it's very fast. Z has no delay.
But why would this be beneficial in a situation where you're expecting, you want to delay to the signals coming from the outside, as if you're not believing them? You think that they might change and it's not good to act too quickly, But why, when they shut off, will you want to stop responding? Because they might realize.
So the question is, why is this useful? Why do we need what's called, this feature is called a sign-sensitive delay. There's a delay when you add the signal, and there's no delay when you take it away. It's a little bit like the cell phone. You need to press for a long time, let's say, to turn it on, short time to turn it off, or something like that. Or my favorite example is in the elevator. So what I want to say is that this function is also used in engineering often, because often the signals, the constraints on a system are not symmetric. And in the case of a lot of times making proteins is costly, and therefore you want to make sure before you start making them. While stopping to make proteins is not costly, so the cost is different for both
You're
situations.
wasting, you're still producing Y, and you're not getting it at the off step.
So you're not producing either Y or Z. You stop producing them. We'll see in a second after the break a biological example of this where we can discuss it. This property, in the case of AND gates, is delay after on, but not off steps. And we're going to take a nice deep sigh of relief now. And we'll take a 10-minute break and be back in 10 minutes. Properties. It has a delay after an on step and no delay after an off step. And as everybody's walking in, we can take another nice deep sigh of relief. Welcome. So just to summarize this, the C1 FFL with AND gate filters out brief input pulses. And what do I mean by that? If the signal is present for just a brief amount of time, let's say shorter than a few minutes, then the output doesn't see it. Only if it's on for enough time after this delay, you start getting a response. So if the signal is in the environment briefly, we don't see it. We don't see it. This equality doesn't register it as worth responding to. If the signal goes away, there's an immediate stop in making the protein Z. Where does this happen in nature? So we took a look. So I just want to say, it was a question during the period. It's very important. What we did here was look at a feed-forward loop 3-gene circuit, but of course it's inside a big network. So there's many. It's embedded inside many other interactions, and those X, Y, and Z can participate in other subgraphs in addition to the feed-forward loop we're dealing with. So we have to do experiments on the living cell where this feed-forward loop is inside its natural context with all the interactions. We go into it and out to it. So we can test ideas like this. It's very important to do experiments. And to do that, you need to choose an example for the feed-forward loop. And Shmulik Mangan, as a graduate student, shows one of the best studied feed-forward loops, or best studied systems, let's say, in E. coli, and noticed that it's designed as a feed-forward loop. This system eats a sugar called arabinose. This is a sugar that E. coli can grow on. And it has two signals. One is this arabinose, that's SY. And SX is a small molecule that E. coli produces within the cell when there is glucose starvation, when it's starving. So the way this works is, if I'm starving and there's arabinose around, and make the proteins that enable me to eat this sugar. So these proteins cut up this sugar into pieces that E. coli can use, and it regulates other genes that are the pumps that pump in this sugar. So that's the logic of the system. And to analyze the system, you can compare it to a simple regulation situation when there is no feed-forward loop. So E. coli provided us with a perfect example. The same signal, SX, same CRP, same protein, regulates another gene. This gene eats a different sugar, okay? But there's no feed-forward loop here. There's also the lac repressor. People here from biology know. So we had to add IPTG to neutralize the lac repressor, those of you that know about that. But you can compare this same X regulates one gene in a feed-forward loop and another gene without a feed-forward loop. So you can compare the dynamics of these two when you apply the signal, which is glucose starvation. You make the cells starve and see at what rate they make these proteins. And how do you see at what rate they make these proteins? By using this green fluorescent protein trick or technology, making the bacteria turn green in proportion to the rate of production of LAC-Z in one kind of bacteria and RBAD in another kind of bacteria by gluing the gene that comes from jellyfish into the DNA of E. coli, connected to the promoter that controls LAC-Z in one strain, and connected to the promoter that controls RBAD in another strain, and compare them inside the automated fluorimeter that measures fluorescence once every few minutes. And you have nice signals. And so this is rate of GFP production, or Z production, is a function of time. And at this point, t equals 0, you add the signal, Sx. You can add cyclic MP outside of the cells. It mimics glucose starvation. Just add the signal with the pipette, time t equals 0. And this is LAC-Z. Oh, yeah, and this is so there's a delay of about 20 minutes. So this is like an on-step of Sx. Did I explain myself? So you see this delay here. It's 20 minutes. And when you take away the signal, there's no delay. The GFP just dilutes away with the cell growth. So we can see that in this system, it's a sign-sensitive delay element. Its parameter is 20 minutes. By the way, E. coli in its natural environment has these spurious pulses of cyclic AMP. It has false positive signals whenever it transits from one kind of growth condition to another, as it moves in our body, let's say from the stomach to down the intestines. It makes these pulses of cyclic AMP, and they last about 20 minutes. That's a fact from other studies. So it looks like evolution has tuned this system not to respond to pulses shorter than 20 minutes or less, corresponding to the ecologically expected pulses. It's very important not to delay when you turn off these signals, because it costs growth rate. E. coli grows slower when it makes unneeded protein. So E. coli tries very hard not to make unneeded protein, and it's very important to protect from making proteins. And when you want to stop making proteins, you don't need a delay. That's no problem. So it's an asymmetric situation. So in this system now, even though it's inside the cell with a lot of other interactions, this circuit appears to have autonomy. That is to say, we can analyze it mathematically as if it was just 3 genes. And it works that way also in the context, or at least it has the function, has this dynamical feature of sign-sensitive delay also inside the full cellular context. So that's the kind of experiments you can do. With this kind of approach, what you can do is you look at the network, you find subgraphs or circuits that occur. You mathematically, graphically analyze what kind of function they can do that simpler circuits can't do. This is actually the minimal circuit, smallest number of components that can do sign-sensitive delay. You can do sign-sensitive delay also with 4 components or 5 components, but this is the smallest circuit that can do it. And then you can experimentally test it by finding an example of that circuit inside the living cell and do experiments on you can measure the input-output relationship inside living cells and test your hypothesis. And of course, you have to test it on each system, because it could be that a feed-forward loop somewhere is actually connected in such a way that it does something else. That's always a possibility, right? But now that we see feed-forward loops and across organisms, you find this network motif is a huge network motif across in the transcription networks of all known organisms. We have an hypothesis of what it does, even if it's in a system we don't understand. And of course, experiments can tell us surprises, new functions, etc. But we have a kind of building an alphabet or a language of circuitry to understand complicated networks. Now, we talked about the AND gate. What happens if it's not an AND gate but an OR gate? That's also a possibility, right? So we can try to analyze that. OR gate, coherent type 1 feed-forward loop. So our circuit is now, instead of an AND gate, it's an OR gate. So either X or Y is enough to activate Z, right? And we can.
It's something before
Oh, I think.
Why does it need both X and Y biologically? It's a matter of concentration, or?
So the question is, biologically, how do you make an AND gate? Is that the question? And so this goes into the molecular details of. It'll be great if someone can close the door. Toda, Lydia. And this is a there's great deal of knowledge about exactly the way that activators and repressors work, and each one works in a slightly different way. So for example, in the a lot of times what happens is that RNA polymerase, the molecule that starts transcription, has with it some factors, or in the case of bacteria, it has several subunits. And sometimes what you need is one of these transcription factors touches one subunit, and another one touches another subunit, and only when both of this happens, RNA polymerase is built like a motorcycle. But you need both. So that's like a molecular thing. Or in eukaryotic cells like ours, has many factors around it, and it's one of those cooperative effects. So this is inside the molecular details.
It's very possible that one of the positive regulators just removes a constitutive negative regulator. So they don't need to actually.
Thank you. So the comment was that one of the positive regulators can kick off a negative blocker that's there, maybe a histone, or not a histone, a nucleosome. And here in my course, by the way, I'm making a choice how deep to go into biological molecular details. And my choice, as you notice, is to minimize it. Of course, I welcome the questions very much. This is easy to find in textbooks on biology. It's very rare to find this way of looking at biology, where you start from the concepts. So that's. But I want you to know that if you're interested in that question, and you look at even in Wikipedia activators, transcription activators, you'll find wealth of information with molecular detail about how they bind in bacteria, and it was beautiful work on the mechanics of it. On this level of description, we're chunking all that molecular detail into things like AND gate, threshold, and then we can say things about the dynamics that don't require us to know all the details in particular. So the OR gate. We'll do the on step. Y, what happens when there's an on step? Y starts being made. What happens to Z? starts immediately, no delay, because it's an OR gate. It's enough to have Y. Thank you. You see, people here are awake. I'm very happy. It's enough to have X. Y doesn't have to cross the threshold. When it crosses the threshold, great, but we're already on. Because OR gate is 1 is enough, either. That's the OR gate. What about when we take the signal away? Y starts going down. What happens to Z? There's a delay. Why is there a delay? Indeed, X star went down, went away, because the signal is away. But it's an OR gate. We still have Y. Thank goodness we have Y. It's like an emergency battery. We have it. It's like when we have this uninterrupted power source in our computer. You know, when the electricity goes down, we still have the battery. If we have an uninterrupted power source, it's around for a while. So Y is around for a while. When will Z stop being made? When we cross this threshold, our famous threshold. And now Y will stop being made. So this is the delay. So OR gate is a sign-sensitive delay for off steps. So it's the same function, but on changes to off. That's what's happening with the OR gate. And indeed, this happens biologically. And again, tested experimentally, you find a circuit that has an OR gate. And this happens when bacteria make their flagellar motor. That is to say, when they've starved and they want to swim after food, they make about 30 kinds of proteins that self-assemble into an electric motor that spins at 100 hertz and a propeller that pushes the cell at 30 times its body length per second, 30 microns per second, and a little computer called the chemotaxis system tells it where to go. It goes up gradients of food, down gradients of toxins. And this decision to make these motors is carried out by a coherent type 1 feed-forward loop. Here's the autoregulation, by the way. This has signals of stress. It's time to move on. The signal here is the motor is halfway made because this transcription factor has an inhibitor sitting on it, inhibiting it. And when the motor is halfway made, this inhibitor flies out. So this is an ingenious way to see if the motor is halfway made. And only when it's halfway made, this one starts, Fly-C is the protein that makes the flagella, the propeller. So when the motor is halfway made, it starts making the propeller, which goes out of this motor and self-assembles into a hollow tube made of monomers that rotates and pushes the cell. But if you make it too soon, it has no way to go out. It's toxic to the cells. There's a pretty ingenious mechanism to make sure that you assemble a motor. And here it's a sum. Actually, it's the sum of these two. Sum is similar to the OR because either one alone can activate. And in this system, again, when you turn this on and connect green fluorescent protein here, you see that when you turn the system on, there's no delay. When you turn the system off, there is a delay. Compared to what? Compared to a strain where you erased Y. So when there's no Y, you can do that. You can erase this gene. As I said, you can read and write into equal. You can cut off this gene. And then it's just a simple situation. And so you can compare what the feed-forward loop is doing to a situation where there's no feed-forward loop. Or you can erase the binding site of A in the promoter of Fly-C. You can also erase this and get rid of this arrow. This is even better, which is in fact what was done in this experiment. And so this delay, by the way, in this case is 1 hour. And again, when you think about why 1 hour, when you measure in E. coli in these conditions, how long it takes to produce a motor, it takes about an hour. And so this motor is produced by parts that assemble like ring, rods, one after the other. And the logic here is that when you're swimming and you reach a situation which is no longer stressful, you don't stop immediately making the motors. You continue making them. And in fact, you make them, and you're able to complete the last motor that you made. And then you shut off. So it's a kind of a regulated self-assembly process in this case. So you can do pretty sophisticated analysis on that. So we see that whereas the AND gate and the OR gate coherent type 1 feed-forward loops are both delay elements, they have delay only in one direction, on steps or off steps, and that's determined by the logic gate. By the way, what do you think this positive autoregulation does for this circuit? Enhances the production of the motor. How? So you can make more fly A, but you could do that without this positive autoregulation. Just make the promoter stronger. We said that positive autoregulation causes a delay. Increases the delay. And here we see an example of one network motif, positive autoregulation, inside a bigger network motif. And this network motif can work autonomously. It enhances the delay here inside the delay done by this network motif. So this 1 hour, instead of 20 minutes, is aided by this positive autoregulation motif inside the bigger motif.
And that's
So
because it involves a repressor.
this is a positive autoregulation. It's all positive.
Yeah, but it's you said that in fact it's
Slows the response time. You can say. The negative autoregulation speeds response times. Positive autoregulation slows response time.
So the autoregulation of delay maintains the delay in the offstep even further
Yeah,
to make sure that whatever motor began to be built.
yeah. That's at least the story that we can, as human beings observing the system, can make, and leads to experimentally testable hypotheses. So you can now, you know already two circuits, feed-forward loops, negative autoregulation, positive autoregulation. You can start making more complicated circuits. Just like E. coli. What about the incoherent feed-forward loop? What about this interesting circuit? Huh? Why should X make Z and make a repressor of Z?
You want to translate this one?
What's the deal here? So when we analyze this circuit, and we're going to do it right now because you're already so skilled, you know how to do this graphically. I guarantee you, you can do it by yourself. And so let's imagine we turn on this X. What happens to Y? Starts being made, right? What happens to Z? So in the beginning, there's no repressor. And there is an activator. There's not much choices here. What happens to Z? Starts being made. Now, when do things start getting interesting? Wait. Huh? When Y crosses its threshold here, K, Y, Z. So this point is interesting. What happens here? The production rate of Z goes down because the repressor binds. It can either go down to 0 or to a lower value. Depends on the design, right? But it goes down for sure. So what happens to Z? Starts going down. It kind of goes down exponentially to its new steady state level, either to 0 or to something else.
Why is it going down?
Because here, at this point,
Maybe it's slowing down.
well, in the exercises, exercise 1, you solved what happens when the production rate of a protein goes from beta to beta prime, something smaller. That point.
That's related to the relations between Y and X, between the effectiveness of the repressor and the...
But for sure, once the repressor binds, production rate is lower. That's for sure, because it's a repressor.
Yeah, but the rate of
The rate is, yeah, the rate depends. By the way, what's the response time here? 1 over alpha. It just depends on the lifetime. And where it reaches is beta prime over alpha y. These are all.
But you don't know the point that Z reached to.
Yeah.
Is already is higher than the new steady state.
Good point.
Is this still lower?
Good point. It could be, right, that goes at a fast rate here and then a slower. That's possible also. That's correct. So it doesn't have to be a pulse like that. It could be also. That's correct. Correct. Good point. So this is a pulse generator. And I just want to make a point here that if we had no, it's also, I also want to make the point, it's also, it's also another way to speed response times. What do I mean by that? If we're going here to the new steady state, if I wanted to design simple regulation with same, so the steady state here, of course, is beta over alpha, same z steady state, then it would look like this. Our halfway point is shorter with the incoherent type 1 feed forward loop. Because, why is that, just intuitively? What you can have here is a very strong production rate, and then after a while, you stop it. It's similar to the negative autoregulation, but it's a different circuit. It's a very good way to make pulses. You see. Yeah.
Explain why does Z have to go down? Why doesn't just the slope of it going up is?
Well, so the question, if I understand correctly, was already asked, and I didn't explain myself. It's possible. It's possible if this is the final steady state, and I stop or y binds before you go higher than this, then you can have two slopes to reach that steady state like that. That's correct. And that'll still be faster than simple regulation reaching that steady state. And depending on parameters, you can have an overshoot. So either way. And in order to design properly, you want this delay to be big enough. Delay here, it's the same. This is the delay, just like in the coherent feed forward loop, to be big enough in order to overshoot. But again, and the shape of this overshoot also depends on the details. If it's a steep regulation function or not, you can have smooth, less smooth, or. But what survives always is this response time, because you can have fast response and then you slow it down, just like fast accelerator and brake. And now you know enough in order to figure out the following circuit. So integrated. What? Yeah? Here I'm going to give an example. This will be the last thing we do. To show you how you can build out of these elements something like, just like you can build out of transistors, you know, a laptop, a cell phone, etc. You can combine these to make interesting dynamics, and this will be our example. This, the biology here is the sporulation of the bacterium. This bacterium, unlike E. coli, when it's starved, it makes a spore that's extremely durable. It can be in the ground for years, decades. And when you put it back into a nice condition, it makes bacteria again. And in order to do that, there's a mother cell, splits its DNA, puts one part in this protected, makes a lot of proteins. They're protection proteins, and then commits suicide. Laying the egg, you could say. In order to do that, it has to make massive waves of proteins to protect this DNA, to cover it with nourishing layers, to make a very tough cell wall. Waves of proteins have to be made. And the circuit that does this was figured out by the Losik lab in Harvard. And he showed me the circuit. I jumped for joy. Because you see in this circuit a coherent type 1 feed-forward loop with two regulators. Sorry, this is an incoherent type 1 feed-forward loop. Make a coherent type 1 feed-forward loop with an AND gate on this incoherent type 1 feed-forward loop. And these two transfer factors make one final coherent type 1 feed-forward loops. And these are groups of genes that encode the proteins that make the spore. There's more details here, but this is the essence. And this is this is a starvation signal. Here is a second signal that comes from other cells around it, etc. With what I taught you today, you are able to make sense of what's going on here. And I'm just going to carry you through. So when there's starvation, this incoherent type 1 feed-forward loop produces a pulse of Z1. This could be tens of genes. Then there's a delay. This coherent type 1 feed-forward loop waits to see, is the starvation signal there for enough time? If it is, there's a delay after the response. It turns on this transcription factor, master regulator, which participates in this incoherent feed-forward loop, producing a second pulse at a delay. And if the signal is around for enough time, this final coherent type feed-forward loop makes the last set of proteins. So this kind of integrated circuit produces waves of gene expression. They're timed and also disregards transient signals. Each one of these Z1 is actually the same incoherent feed-forward loop is, as I said, many, many genes. And by playing with the parameters, the K, the Y, you can make these waves have different properties for each of the genes. That's fine details. So to summarize, we looked at the E. coli transcription network. We found an overrepresented circuit, this feed-forward loop. We saw that of the 8 possible type of feed-forward loops, 2 are mainly found, coherent type 1 and incoherent type 1. The coherent type 1 can function as a sign-sensitive delay element. It can have a delay after on steps if it's an AND gate, or delay after off steps if it's an OR gate, thereby allowing you to disregard short pulses of on or off signals, acting like a uninterrupted power source or like a delay element. And the incoherent type 1 feed-forward loop activates, but at the same time represses a delay that can generate pulses and speed responses. And bacteria, this is a different bacteria from E. coli, of course, has the same network motifs, can put them together to generate, in fact, arbitrarily complex time courses. You can get as close as you want to any time course by feed-forward loops, coherent type 1 and incoherent type 1. They're like universal in that sense. And you can, of course, connect feed-forward loops in a way that where you can't understand them anymore. But the circuits that have been analyzed so far, they seem to be connected in ways that are possible to understand based on the autonomous function of each of these feed-forward loops on its own. Of course, every system requires its own experimental study. So I hope you leave this class with some tools for understanding gene circuits, graphic tools, and it's a good time to take a nice deep sigh of relief. See you next week.